The next question, some soils may be deficient in nitrate. Suggest how nitrate deficiency could limit the quantity of SPPS made by SOX4 plant. So this video is new, but the continuation of the previous question, and genetically modified the maize plant, which express more enzymes to make RUBP. SBPase is enzyme, and it can produce more biomass. That is advantage of genetically modified plant. Suggest how nitrate deficiency could limit the quantity of SPPS made by SOX4 plant. Now, for that, what is SPPAS? SPPS is an enzyme. So enzymes are proteins. Enzymes are proteins. The proteins, the code by the gene, SBPS gene, make the proteins. And then this gene is transcribed to messenger RNA, and the messenger RNA translation make the proteins. So nitrogen needed to produce nucleotide. So nucleotide which contain nitrogen, carbon, hydrogen, oxygen, nitrogen, and DNA molecule and RNA molecule, phosphorus and nitrogen. And also nitrogen needed to produce amino acids. So both DNA and messenger RNA, they produce, to produce those things, they need the nucleotide. Nucleotide production required nitrogen, and amino acid make the protein and enzyme, it also need nitrogen. So answer is, again the model answer, nitrogen is essential for the production of nucleotide, which in turn are necessary for the transcription of the SBPase gene into mRNA. Additionally, nitrogen is required for the production of amino acid, subsequently enabling protein synthesis and enzyme production. So this is a new question. An experiment was carried out to investigate the effect of changing light conditions on the pH of the chloroplast stroma. Scientists followed pH changes in chloroplast stroma using fluorescent chemicals that can be used as pH indicators. Chloroplasts were isolated from cells. A suspension of chloroplasts were prepared and kept in the dark for 180 seconds. The chloroplasts were exposed to a period of light of fixed intensity for 240 seconds, then returned to dark conditions. The pH of chloroplast stroma was continuously measured and recorded. Figure 3.2 shows the result of this experiment. So 0 to 720 pH of chloroplast stroma on y-axis, 7.2 to 7.7, the highest value, dark, light, dark. First dark, and then a light, and again a dark. So we can see during the dark, pH is between 7.4 and 7.3. And when light increase, pH increase to 7.6. And again, when dark, pH decrease. Describe the results shown in figure 3.2. So I draw a line to show the dark and light, and the pH here, it's between slightly changing the pH between 7 and 7.3 here, and then pH increase to 7.6, and then pH decrease in the dark. So these points I consider, here is the lowest, highest, highest, lowest. The pH level is greater in the presence of light compared to when it's dark. So I just make the overall result. It is the pH is higher in the light and low in the dark. When light is introduced, the pH sharply increases, and in the presence of light, the pH stabilizes. So that means pH keep higher in the presence of light. Conversely, when returning to the dark, the pH gradually decreases without necessarily returning to its original dark pH level. So this is a model answer. I just add the technical terms and the correct way of writing to describe the result. The pH level range from 7.31 to 7.6 in the dark conditions, and from 7.5 to 7.61 in light conditions. discuss how the result support that chemiosmosis occur during phosphorylation. So it is, so I'm going to show you this. So that's a membrane. So PS2, PS1, 1, 2, 3, 1, 2, 3. So then the light absorb, the electrons are excited and move into this complex. Light absorb, electrons are excited and moving to make NADPH. And then the energy of electrons used to pump hydrogen ion in. Then ATP synthase complex, the hydrogen is going out by chemiosmosis. So that time ATP is produced. The briefly light dependent reaction is this. So here is a chemiosmosis. So then the protons with the higher gradients to lower gradients moving through the ATP synthase complex is the chemiosmosis. During that time, the energy is used to make ATP. That is called photophosphorylation. Photo means energy of the sunlight used to make ATP adding the phosphate group. Here again, it shows this light dependent reaction. And my answer for this question is the stroma exhibit higher pH as a result of the movement of proton hydrogen ions out of the stroma and into the thylakoid space or lumen. Then this process leads to an increased proton concentration in the thylakoid space or lumen. Subsequently, the proton ion diffuse back into the stroma through ATP synthase.